Assalamu alaikum dear students. I hope you all will be fine and enjoying with a good health. I welcome you to the lecture number 45. Here I am, Ms. Sana Ismail, lecturer of botany and working as a co-teacher in the Department of Botany at the University of Education, Lahore. Today I am going to deliver the lecture about polyembryony in different groups of plant from the subject plant systematics, anatomy and development. The course code of this subject is BOTN 1112. Let's begin our lecture by discussing the outline of today's lecture. So here is the lecture outline. In this lecture, first of all, we discuss about polyembryony in different groups of plants, such as in the gymnosperms, and also discuss about polyembryonic process in the angiosperms. Then we discuss about the factors that affecting the polyembryony and the factors which causes the polyembryony. Then we have the inheritance of nuclear embryony, their distribution of nuclear embryony, and then we discuss about the degree of polyembryony and their significance of polyembryony. At the end, the references are present which are used to prepare this lecture. Polyembryony in plants. Polyembryony is termed as the production of two or more than two embryos from a single seed. It is a type of apomixis during the autonomous development of supernumerary embryos, which takes place in the seed, and as a result, the genetically similar progenies are developed. Polyembryony has great importance in generation of true quality plants. It is exhibited by a number of plants, yet little is known about the origin of their reproductive phenomena. To understand the origin and formation of multiple ovules in plants, we will discuss it in angiospermic seeds and gymnospermic seeds to understand their polyembryonic process in this lecture. Let's discuss polyembryonic process in different groups of gymnosperms one by one in the next slide. Polyembryony in cycadels. In cycadels, polyembryony is not a usual phenomenon, but in 1964, Rao reported the occurrence of simple polyembryony in Cycas cercinalis. It is the only gymnosperm species which found among natives of Sri Lanka. In this species, two adjacent archegonia of the same ovules sometimes grow independently into two embryos and also rarely into two seedlings. In 1952, De Silva and Thambia also reported the occurrence of polyembryony. Only one out of the several embryos remains potential and persists in this species. Polyembryony in Coniferous. In case of Coniferous, some polyembryony occurs in the majority of its members, and here the number of embryos varies from 2 to many. It has been reported that cleavage polyembryony has occurred in several groups of Pinaceae, Taxodiaceae, Coniferous, and Podocarpaceae. In Coniferous, both simple and cleavage polyembryony are common. Polyembryony in Taxodiaceae. Various archegonia are present in the female gametophyte of Taxus. Simple polyembryony occurs due to the fertilization of many of the archegonia eggs. But out of many, only a single embryo attains maturity. In 1948, Sterling reported that mostly cleavage of suspensor cells occurs only in Taxus. The suspensor separates from each other, and each of them may carry one or more embryonal units. Sometimes group of meristematic cells are observed at the base of the suspensor cells. These groups of cells are called the rosette embryos. And the further development, however, does not take place in these embryos. So this is all about the polyembryony in Taxodiaceae. Let's discuss about the polyembryony in Genitales, which are also the member of the gymnospermic plant. All Genitales exhibit polyembryony, and polyembryony is found to be of very high order in Genitum. In this group, there are not only several prothelines and zygotes in each seed, but there is the multiplication of embryos from each zygote by the branching of the primary suspensors. There can be also the occurrence of further proliferation of the secondary suspensors. In 1965, Sporny stated that only one embryo normally reaches maturity in each seed. Vassell in 1959 and Maduleta in 1960 have also reported some cases of polyembryony in Genitum ula and Genitum geminum. The embryo development in several species of Genitum has been studied by many different workers, but according to Mashwari and Vassell in 1961 stated that the first division of the zygote in gymnosperms occur, and these are free nuclear division in the zygote. In this respect, a link between gymnosperms and angiosperms are created, and by showing the both free nuclear division as well as the cell division. According to Thompson in 1916, a two-celled proembryo is formed. From each of these two cells develop a tube called a suspensor. Now the nucleus divide and one of the two nuclei undergo free nuclear division and forming the four nuclei. The embryo gets organized by these four nuclei, and there is no division in the other large nucleus. Maduleta in 1960 has worked on the zygote development in Genitum geminum. According to her, 2 to 4 or sometimes up to 12 zygotes may develop in gametophyte, of which normally one remains functional. From the zygote develop generally one or sometimes 2 to 3 small tubular outgrowths. Only one of these tubes receive the nucleus and survive, while the remaining tube disintegrates and soon die. The surviving outgrowth elongates and becomes branched and grows into different directions through the intercellular spaces of the endosperms. And all the primary suspensors or tubes usually remain coiled round each other. In this figure, it is the primary suspensor. A small cell is cut off at the tip of the primary suspensor tube in genitum genemon. It soon divides first transversely and longitudinally, and resulting into 4 cells. Now irregular divisions take place, forming a group of cells, which is called the embryonic mass. And some of these cells divide and elongate to form secondary suspensors. The remaining cells at the tip form the embryonic. As in case of genitum ola, a small cell is cut off at the tip of the tube called peculiar cell. This peculiar cell soon divides and forms a group of cells. This secondary suspensor and embryo mass are differentiated from this group of cells. The secondary suspensor and embryonal mass are differentiated from this group of cells. By this time, the wall of the tube starts to become thick. Whatsoever may be the pattern of formation of the embryonal mass and secondary suspensor, the cells of the former are small, compact, dense in cytoplasm and develop into embryo proper, while that of the latter, for example these secondary suspensor, are thin-walled, uninucleate and highly vacuolated. The primary and secondary suspensors help in pushing the embryo into the endosperms. Soon, a stem tip with 2 lateral cotyledons form in the tip regions of the embryo mass. On the opposite side, the development of the root tip with a root cap occur. A further development after the formation of stem and root tips, the feeder is a protuberance-like structure which present in between the root tip and the root cap, root tip and the stem tip. Thus, the stem tip, 2 cotyledons, root tips and root cap are the parts of the mature embryo. So this is all about the development of embryo in polyembryony in different groups of angiosperms. The angiosperms show the unique phenomena of double fertilization. The egg after fertilization develops into an embryo. It is nourished in the younger stages by the suspensor and later stages by the endosperms. A post-fertilization tissue resulting from the fertilization of polar nuclei. Besides the egg, other constituent cells of the embryo sac with or without fertilization produce embryos. Moreover, the activity of one or more cells of the ovule outside the embryo sac also leads to the development of accessory embryos, and such embryos are asexual. In addition to the zygote embryo, sexual or asexual non-zygotic embryos may also develop in the same seed, and such condition is referred to a polyembryony. In fruit tree crops, polyembryony is also common and occurs in many crops. In citrus plant, mangoes, Syzygium species, in kiwi, almonds, Frageria species, and peach, the process of polyembryony is present. The occurrence of polyembryony depends on species and varieties. In other words, not all the species of a genus exhibit polyembryony, and not all varieties of a species show polyembryony, and the reason could be genetic. Most of the citrus species show polyembryony, while the Citrus medica, Citrus grandis, Citrus latifolia, and Citrus nobilis are monoembryonic. In mango, most of the varieties grow in coastal area and show polyembryony. So here is the factors that affecting the polyembryony. The number and type of embryo produced may vary from tree to tree and also at different positions on a single tree. The variation has been suggested to be controlled by minor genes, pollen sources, and environmental conditions. If the zygotic embryo does not survive, the development of nuclear embryo is dependent on the initial development of zygotic embryo or the process of fertilization of the ovule, and may also dependent on their pollination. The nuclear embryo apparently may not develop independently of the process of sexual reproduction, and in most of the cases it requires the development of zygotic embryo. In citrus plant, neither the seed setting nor the formation of the nuclear embryo take place without the pollination of flower. The development of nuclear embryo is induced by fertilization process, and it has been established that pollen play a definite role in the formation of additional embryos. The polyembryony is also affected by the type of pollinators, by pollen viability, plant nutrition, temperature, environment, and also affected by soil humidity. The development of endosperm is required for the growth of embryo, since endosperms supply food to developing embryos. The fertilization is prerequisite for the production of matured embryo, and the growth of nuclear embryo is arrested at later stages of development if endosperm is not formed. Therefore, any factor that affects pollination, fertilization, or seed development, it also affects the percentage of polyembryony and embryo number per seed. Causes of polyembryony. Many theories have been proposed to explain the occurrence of polyembryony, but most of them are not sufficiently validated. Haber landed in 1921 to 1922 proposed the necrohormone theory. This theory advocated that the degenerating cell of the nucleus act as a source of stimulus for the adjacent cell to divide and form adventive's embryo. But the theory could not be validated as adventive's embryo could not be induced by damaging the nuclear cells. The monoembryonate condition in some species of citrus has been described through the synthesis and release of certain volatile and non-volatile embryogenic inhibitors in their ovule, which do not occur in the ovules of polyembryonate species. Ethanol and ethylene are important volatile inhibitors that produced by the ovule of monoembryonate species of citrus such as the Citrus medica. The ethylene, among other substances, could be repressing the development of nuclear embryos through its union with an ethylene receptor protein of nuclear cells. The fewer number of adventive embryos in south side of the trees than that of north side may be due to the more synthesis of ethylene, ethanol, and abscisic acid due to variation in sunlight and temperature. And the non-volatile component of the inhibitors was oxons, abscisic acid, and gibberellins. But genetic theory is the most accepted theory as the presence of polyembryony is determined by the gene. Inheritance of tubular polyembryony. Polyembryony in citrus and mango is generally controlled by a dominant gene having heterozygous allele, while homozygous recessive gene is present in monoembryonic citrus species. In monoembryonic species, these recessive genes may synthesize a potent inhibitors of embryogenesis. A variable degree of polyembryony was recorded in polyembryonate offspring obtained by crossing monoembryonic and polyembryonic, and also between the polyembryonic parents, that implied the presence of minor genes affecting degree of polyembryony. Moreover, the presence of modifier or duplicate genes should also be considered, as in some crosses between monoembryonic and polyembryonic parents and between polyembryonic parents. The progeny ratio varied greatly from 1 to 1 or 3 to 1 ratios, and it may be concluded that mostly polyembryony is controlled by heterozygous dominant gene, and the absence of dominant allele lead to monoembryony. But in some of the cases, modifier genes and minor genes are also present, that with the ratio and degree of polyembryony respectively. Degree of polyembryony is the frequency of occurrence in multiple seedling in a seed. It varies with species and varieties, and the variation also occur with location, environment, and position of fruit on tree. On the basis of degree of polyembryony, species and varieties can be divided into 3 categories. The first is slightly polyembryony, in which the polyembryony occurs up to 25%. The second is moderately polyembryony, in which the 50% up to 50% polyembryony occur. And the third one is highly polyembryonic, which is the more than 50% polyembryony are performed. On the basis of number of embryos, the embryony may be divided into different morphotypes. It may be divided into singlet, in which one embryo are present in one seed; duplet, in which two embryos are present in one seed; triplet, the three embryos are present in one seed; quadruplet, in which four embryos are present; quintuplet, in which five embryos are present on per seeds; sextuplet, six embryos are produced in single seed; and heptuplet, in which the seven embryos are present, and up to so on. In apomictic citrus species, mostly the quadruplet, it means four embryos are present in one seed, and triplet embryos mean three embryos on per seeds are more frequently, regardless the genotype. Significance of polyembryony. Polyembryony or nuclear embryony play an important role in horticulture, cytogenetics, and plant breeding. Nuclear embryony helps in producing genetically uniform seedlings of the parental type for better clone. Polyembryony helps in the large-scale propagation of desired genotype. The nuclear seedlings show a restoration of the vigor lost after repeat vegetative propagation. The nuclear embryos are free from diseases, as in vitro nuclear embryony is the only practical approach to raise virus from clones of polyembryonate citrus varieties in nature. Applieds can be used for cytogenetic studies, and the homozygous diploid can be raised from haploid by colchicine treatment. So this is all about for this lecture, and here is the reference that are used during the preparation of this lecture. Hopefully these references are helpful to understand and clear the concept about polyembryony topic. Thank you.